Let me ask you something uncomfortable. You slept 8 hours last night. You ate well this week. You trained, maybe not perfectly, but you showed up. You did what the system told you to do: rest, hydrate, recover. And yet this morning, when you put your feet on the floor and stood up, there was something in your body, a weight, a dullness, a fog, that wasn't there 10 years ago. Not pain exactly, not exhaustion exactly, something quieter than that, something harder to name. And the dangerous part is, you've started calling that normal. That's the moment I want to talk to you about today. Not the dramatic crashes, not the injuries, not the obvious signs that something is wrong. I want to talk about the slow erosion, the almost imperceptible slide from recovery into something that looks like recovery from the outside, but is biologically something else entirely, something that researchers have a specific name for, and almost no one over 40 has been told about it. Because here's what most people in their 40s and 50s believe: that recovery is about rest. That if you give your body time, it will repair itself. That sleep and nutrition are the two main levers. That soreness means growth, and absence of soreness means you're fine. Every single one of those beliefs is incomplete. And for people over 40, some of them are actively counterproductive. What if the reason you feel like you're running on 70%, not broken, not sick, just perpetually below your potential, has nothing to do with how much you're resting? What if the problem isn't that your body isn't getting enough recovery time, but that the biological machinery responsible for executing recovery has been quietly degrading for years, and the signals your body uses to coordinate repair are now arriving distorted, delayed, or not at all? What if surviving and recovering look exactly the same from the outside, but inside, only one of them is actually rebuilding you? That is the question this video is going to sit with. Not answer in the first 60 seconds. Sit with. Because it deserves to be understood before it gets answered. This is part 4 of the Recovery Science After 40 series. If you're joining for the first time, welcome. You don't need to have seen the previous parts to follow this one, but I do recommend going back because we've built a foundation that makes everything here land with more weight. And to those of you who have been here since part 1, you already know that what we do on this channel is different. We don't give you 5 tips. We don't hand you a checklist. We explain the biology in full so that when you make a decision about your body, you actually understand what you're doing and why. All protocols on this channel are reviewed by a clinical pharmacist and researcher, and all scientific references and sources used in this video are listed in the description below. Every claim has a citation. Every recommendation has a mechanism. That is not optional here. Now, let's go back to that moment this morning, when you put your feet on the floor. For most men and women over 40, that moment has changed. It used to be instantaneous. Body on, system ready, no negotiation required. Now there's a delay, a negotiation, a quiet tax your body collects before it lets you start the day. And you've probably adapted to it so well that you no longer even notice you're paying it. But your cells have noticed. Your mitochondria have noticed. Your inflammatory signaling has noticed. Your hypothalamic pituitary axis has noticed. Every system responsible for coordinating recovery has been quietly keeping score, and what they've been recording is not recovery. It's compensation. Compensation is not the same as recovery. Compensation is what your body does when it can no longer recover properly. It adapts to the deficit. It learns to function in the damage. It gets better at appearing okay while the underlying architecture continues to degrade. And the cruelest part of this process is that it's gradual enough that you blame yourself. You think you're getting older. You think this is just what 45 feels like. You think you need to try harder, sleep more, stress less. But the real issue is not discipline. It's not mindset. It's not even sleep quality in isolation. It's a cascade of biological failures that have been building for years, and they all have names, mechanisms, and increasingly, solutions. Here's the question I want you to hold as we move through this video. What if the most dangerous sign that your recovery has broken down isn't how you feel after training, but how you feel when you're not training at all? Don't answer that yet. Let the question settle. Because what comes next is going to change how you read your own body. Permanently. The word recovery is one of the most misused words in health and performance. Most people use it as a synonym for rest. They are not the same thing. Rest is passive. Recovery is an active biological process, a coordinated, energetically expensive cascade of molecular events that requires specific hormonal signals, adequate cellular fuel, functional mitochondria, and a precisely calibrated inflammatory response. When any one of these elements is missing or impaired, the cascade doesn't pause. It distorts. And a distorted recovery process produces something that feels almost like recovery, but isn't. To understand why recovery after 40 is fundamentally different from recovery at 25, you need to understand what happens at the cellular level immediately after a physiological stressor. Whether that stressor is training, sleep disruption, emotional stress, a period of caloric restriction, or even a long workday. The moment your body registers that stressor, it initiates what's called the damage-associated molecular pattern response, or DAMP response. This is your body's internal alarm system. Damaged cells release signaling molecules, including HMGB1, heat shock proteins, and mitochondrial fragments, that tell the immune system, something has been disrupted, send resources. This is normal. This is good. This is the first phase of repair. In response to these signals, your immune system activates a precisely orchestrated inflammatory response. Cytokines, chemical messengers including interleukin-6, interleukin-1 beta, and TNF-alpha, are released. These molecules don't just cause inflammation, they coordinate it. They signal macrophages to clear debris. They stimulate satellite cells in muscle tissue to begin differentiation. They activate the processes that ultimately lead to adaptation and growth. Here's where age changes everything. In a young body, this inflammatory cascade is sharp, specific, and self-limiting. It rises, does its job, and resolves within a defined window, typically 48 to 72 hours for moderate physiological stress. The resolution is not just the absence of inflammation. It requires an active anti-inflammatory process, the production of specialized pro-resolving mediators, or SPMs, including resolvins, protectins, and mycins, lipid compounds derived from omega-3 fatty acids that literally instruct the immune system to stand down and switch into repair mode. After 40, this resolution process slows, significantly. The research is unambiguous on this. A landmark 2019 study published in Nature Aging demonstrated that aging is associated with a progressive impairment in the production and activity of SPMs, meaning the inflammatory signal goes up on schedule, but the off switch arrives late, sometimes very late, sometimes it barely arrives at all. This produces a state that researchers now call inflammaging, a portmanteau of inflammation and aging, characterized by low-grade, chronic, systemic inflammation that never fully resolves between recovery cycles. You are perpetually in a mild inflammatory state. Not enough to make you clinically sick. Enough to impair everything. And here's what inflammaging feels like from the inside. Because this is the part no one explains clearly. It feels like stiffness that doesn't fully resolve in the morning. It feels like mood that is slightly flat. Not depressed, just not elevated. It feels like cognitive clarity that is 90% of what it used to be. Enough to function. Not enough to feel sharp. It feels like motivation that requires activation energy it didn't used to require. It feels like workouts that don't give you the same return, the same effort, less result. It feels like a body that is perpetually at 75%, regardless of how much you sleep or how well you eat. Sound familiar? But here's what makes this truly important, and what most physicians never explain. Inflammaging is not just a consequence of aging. It is also a driver of it. Chronic low-grade inflammation accelerates the very mechanisms that cause further dysfunction: mitochondrial degradation, telomere attrition, insulin resistance, hormonal axis suppression. It is both symptom and cause, a feedback loop that tightens over time unless something specifically interrupts it. Now, you might be thinking, okay, inflammation is the problem. I'll take anti-inflammatories. Reduce the inflammatory load. That's the obvious conclusion. And it is almost entirely wrong, because the goal is not to suppress inflammation. The goal is to restore resolution. These are different interventions with completely different outcomes. Suppressing inflammation blunts the repair signal. Restoring resolution allows the signal to complete its job and disengage correctly. But here's where the mechanism gets deeper. And this is the part that most people, including many practitioners, have never been shown clearly. The problem is not just that inflammation doesn't resolve on time. It's what that delayed resolution does to the second major axis of recovery. The one controlled not by your immune system, but by your endocrine system. Specifically by an axis that most people know by name, but have never fully understood in function. And once you see how these two systems interact, or fail to, you will understand exactly why surviving and recovering look identical from the outside, but produce completely different outcomes inside. The next layer of this goes somewhere most people have never looked. The hypothalamic pituitary adrenal axis, the HPA axis. You've probably heard the phrase. You've probably heard it connected to stress, cortisol, burnout. What you almost certainly haven't heard is what happens to this axis specifically after 40, and how its dysfunction is directly tied to the failure of recovery resolution we discussed in the last block. Let's start at the top. The hypothalamus, a small region at the base of the brain, is the master coordinator of your body's stress and recovery response. It doesn't just regulate stress hormones. It integrates signals from virtually every major system in the body: immune, metabolic, endocrine, circadian. It is the central command for biological adaptation, and it communicates with the rest of the body primarily through a chain of hormonal signals that cascade down through the pituitary gland to the adrenal glands, the gonads, and the thyroid. Here is what's critical to understand. The HPA axis does not operate independently. It operates in constant dialogue with the inflammatory system. Specifically, the inflammatory cytokines we discussed in Block 2, IL-6, IL-1 beta, TNF-alpha, are not just immune signals. They are also potent neuroendocrine modulators. They directly influence hypothalamic function. And in the context of inflammaging, where these cytokines are chronically elevated at low levels, they produce a specific and consistent pattern of hypothalamic dysregulation. That pattern is called HPA axis blunting, and it is arguably the most underdiagnosed and underappreciated mechanism of age-related performance decline in existence. Here's what blunting means mechanistically. In a healthy young HPA axis, the cortisol response to a stressor is rapid, proportionate, and followed by an equally rapid return to baseline. This is called a robust cortisol awakening response, or CAR, and it is one of the most reliable biomarkers of HPA axis health that we have. The CAR is the morning cortisol surge that should occur within 30 to 45 minutes of waking. Its job is not to stress you. Its job is to prepare every tissue in your body for the demands of the day, mobilizing glucose, priming immune function, activating the cardiovascular system, sharpening cognitive circuits. In individuals with blunted HPA function, the CAR is flattened. The morning surge either arrives late, rises inadequately, or doesn't sustain long enough to complete its preparatory role. The result is a body that begins every day slightly under-resourced, tissues that are not fully primed, cognitive systems that are slow to activate, immune function that is not properly calibrated. Now think about what that means for recovery specifically. Recovery depends on your body accurately reading the damage signal, the DAMP response, and coordinating the appropriate resource allocation to repair it. That resource allocation is mediated significantly by cortisol, not through suppression of the immune response, but through its role as a metabolic choreographer, timing the delivery of glucose and fatty acids to repairing tissues, modulating the activity of satellite cells in muscle, regulating the rhythm of protein synthesis. A blunted cortisol response means that even if all the other elements of recovery are in place, the coordination signal is impaired. Resources don't arrive in the right tissues at the right time. The repair process begins, but proceeds inefficiently. And here's where it becomes recursive, because this is the part that creates the loop most people are trapped inside without knowing it. The inflammatory cytokines that blunt HPA function are themselves exacerbated by poor recovery. Incomplete recovery, recovery that goes through the motions but doesn't complete the biological cycle, leaves residual cellular damage. That residual damage sustains the cytokine signal. That sustained cytokine signal further blunts HPA function, which further impairs recovery, which produces more residual damage. This is not a metaphor. This is a documented biological feedback loop, and it tightens with every cycle of incomplete recovery. But there's a third actor in this system that most recovery discussions never mention. It sits at the intersection of the immune system, the endocrine system, and the circadian system, and its function changes dramatically after 40 in ways that explain a great deal about why nighttime recovery is so different from what it used to be. The receptor dynamics involved are sophisticated, the downstream signaling cascades are specific, and once you understand them, you will see exactly why the standard advice, sleep more, stress less, eat clean, is targeting symptoms, not the mechanism. The actual target is something else entirely, and it's been hiding in plain sight in the research literature for over a decade. What comes next in Block 4 will connect the final piece of this picture, and it involves a molecule you almost certainly have in your bloodstream right now, at levels that have been quietly declining since your mid-30s, driving almost every symptom you've normalized. There is a molecule that sits at the intersection of sleep, inflammation, immune regulation, endocrine function, and cellular repair. Its name is melatonin. And before you dismiss that word, before your brain files it under sleep supplement not relevant, I need you to understand that what melatonin actually is and what it actually does in the body has almost nothing to do with how it's marketed or discussed in mainstream health media. Melatonin is not primarily a sleep drug. It is a pleiotropic signaling molecule, meaning it has biological effects across multiple organ systems simultaneously, and it is one of the most potent endogenous antioxidants ever identified. Its role in recovery is not peripheral, it is central, and its production declines by approximately 10% per decade beginning in your late 20s, with a particularly steep drop after 40, and an even steeper drop after 50. Let's follow the mechanism precisely, because the pathway here is what makes this relevant to everything we've discussed so far. Melatonin is synthesized in the pineal gland from serotonin, in a process that is directly regulated by light exposure through the suprachiasmatic nucleus, the master circadian clock in the hypothalamus. Notice that word, hypothalamus, the same region we discussed as the coordinator of the HPA axis. These systems share real estate at the highest levels of neuroendocrine control. They are not parallel systems that happen to interact occasionally. They are deeply integrated, and disruption in one propagates through the other with predictable consequences. During deep sleep, specifically during the first and second cycles of slow-wave sleep, melatonin reaches its peak concentrations. And during those same windows, it is performing a set of functions that are rarely discussed together. First, it directly scavenges reactive oxygen species, free radicals generated during the day's metabolic activity, at the mitochondrial level. This is not incidental antioxidant activity. This is targeted mitochondrial protection occurring during the precise window when mitochondria are most vulnerable to oxidative damage. During the transition from high metabolic demand during waking to the repair-oriented metabolism of deep sleep. Second, melatonin directly modulates the inflammatory response. Specifically, it inhibits NF-κB, nuclear factor kappa light chain enhancer of activated B cells, one of the most important transcription factors controlling inflammatory gene expression. When NF-κB is active, it upregulates the production of pro-inflammatory cytokines, including the same IL-6 and TNF-alpha we discussed earlier. Melatonin's inhibitory effect on NF-κB is therefore a direct brake on the inflammaging process. It is, in part, one of the biological reasons why young people recover faster. Not just because they have more melatonin, but because that melatonin is actively suppressing the chronic inflammatory signal during exactly the window when cellular repair is most active. Third, and this is where the clinical data becomes especially striking, melatonin directly stimulates the production of growth hormone-releasing hormone from the hypothalamus. This means that declining melatonin levels in individuals over 40 are not just associated with worse sleep. They are mechanistically linked to the well-documented reduction in growth hormone pulsatility that occurs with age. Less melatonin means less nocturnal GH release, means less protein synthesis, less fat mobilization, less IGF-1 production, and less activation of the satellite cell proliferation that is essential for muscle repair. In 2021, a comprehensive review published in the Journal of Pineal Research synthesized the data across 43 studies examining melatonin's role in musculoskeletal recovery. The conclusion was unambiguous. Melatonin deficiency significantly impairs multiple independent pathways of recovery, not through a single mechanism, but through the simultaneous degradation of mitochondrial protection, inflammatory regulation, and anabolic signaling. These are not downstream effects. These are primary mechanisms. Now think about what that means experientially, because the reason this matters is not academic. It's lived. Think about the following symptoms and ask yourself honestly how many apply to you. Waking between 2 and 4 in the morning regularly. Feeling a second energy wave after 9 or 10 p.m. that makes it harder to fall asleep. Waking in the morning feeling like your sleep didn't fully land. Like you went through the motions of 8 hours but didn't get the restoration. Slower recovery from training over the past 3 to 5 years, even when training volume has stayed the same or decreased. Inflammation that seems to linger. Mild cognitive fog in the late morning before your first stimulant. A mood baseline that sits at neutral rather than positive, requiring activation. These are not randomly clustered symptoms. They are the expected clinical presentation of declining melatonin function in the context of an aging HPA axis and chronic low-grade inflammaging. They are connected. They share a mechanism. And that mechanism can be addressed, but almost never in the way it's being addressed by the people who are struggling with it. Because here's the thing most people do when they identify melatonin as a factor. They take the wrong dose, at the wrong time, without addressing the upstream light exposure problem, without supporting the serotonin pathway that melatonin requires as its precursor, and without accounting for the mitochondrial dysfunction that makes the entire signaling cascade less efficient, regardless of how much melatonin you supplement. And that gets to the next layer, the deepest one in this series. Because understanding the role of the mitochondria in recovery after 40 is what separates a protocol that helps from a protocol that transforms. What comes next is the mechanism most recovery discussions never reach. But it's the one that changes everything. Everything we have covered so far. The inflammaging loop, the HPA blunting, the melatonin cascade, the disruption of nocturnal anabolic signaling. All of it leads to the same place, the mitochondria. Not because mitochondria are a trend, not because biohacking culture has elevated them to a convenient scapegoat, but because the mitochondria are genuinely where recovery happens or fails at the most fundamental level of cellular biology. And after 40, the decline in mitochondrial function is not a background variable. It is the central variable. Let's establish the mechanism precisely. Mitochondria are not static organelles. They exist in a constant state of what biologists call mitochondrial dynamics, a continuous cycle of fusion and fission. Fusion is the process by which mitochondria merge, pooling their membranes and contents, sharing resources and distributing damage across a larger network. Fission is the process by which damaged mitochondria are isolated and prepared for elimination through mitophagy, the selective autophagy process that removes dysfunctional cellular components. The health of this dynamic cycle is one of the most important determinants of cellular recovery efficiency. After 40, the balance between fusion and fission begins to shift in a specific and consistent direction, toward fission. This is driven in part by the same chronic inflammatory cytokines we discussed. TNF-alpha specifically has been shown to directly activate DRP1, the primary protein responsible for initiating mitochondrial fission. When DRP1 is chronically hyperactivated, the mitochondrial network fragments. Instead of a coordinated, interconnected network capable of efficient energy production and resilient response to oxidative stress, you are left with isolated, poorly communicating islands of mitochondria, each individually more vulnerable, collectively less efficient. This fragmentation has direct consequences for recovery. Specifically, It impairs what is called oxidative phosphorylation efficiency, the process by which mitochondria convert the electrons from food into ATP through the electron transport chain. In fragmented networks, the electron transport chain develops what researchers describe as electron leak, electrons jumping off the chain before they complete the oxidative phosphorylation process, and instead combining with oxygen to produce superoxide, the reactive oxygen species that, at elevated levels, becomes a primary driver of cellular aging and tissue damage. Here is the experiential translation. Your body is producing less energy per unit of metabolic fuel. You are eating, you are sleeping, you are resting, and your cells are still generating less ATP than they were 5 years ago, while simultaneously generating more oxidative damage. The effort you put in is not fully converting to the output you need. And the damage from each session of training, work, or stress is not being fully cleared before the next cycle begins. This is the biological definition of surviving rather than recovering. The damage accumulates faster than the repair can keep pace. Not dramatically, not in a way that breaks anything acutely, but progressively, in increments too small to notice day to day, large enough to register year to year. Now here is where peptide science enters the picture. And this is where part 4 of this series diverges from anything we have discussed in the previous episodes. There is a growing body of clinical and preclinical research examining the role of specific peptides in restoring mitochondrial dynamics. These are not generic peptides. These are molecules with defined mechanisms of action at the level of mitochondrial biogenesis, fusion-fission regulation, and mitophagy optimization. The most researched in this context include SS31, also known as elamoprotide, a mitochondria-targeted antioxidant peptide that localizes specifically to the inner mitochondrial membrane and stabilizes cardiolipin, the lipid that anchors the electron transport chain complexes and maintains their functional organization. In both animal and early human studies, SS31 has demonstrated the ability to reduce electron leak, improve ATP production efficiency, and attenuate the structural fragmentation of the mitochondrial network. A second peptide of significant interest is MOTSC, a mitochondria-derived peptide encoded within the mitochondrial genome itself. MOTSC acts through AMPK activation, a pathway that is central to mitochondrial biogenesis, glucose metabolism, and inflammatory regulation. Importantly, MOTSC levels decline with age. And in early human studies, MOTSC supplementation has been associated with improved metabolic flexibility and reduced markers of systemic inflammation. And then there is the connection to BPC-157 and its documented influence on the nitric oxide system. Relevant here because nitric oxide is a critical regulator of mitochondrial biogenesis through its activation of PGC1-alpha, the master transcriptional regulator of mitochondrial genesis and oxidative capacity. When BPC-157 upregulates nitric oxide availability through eNOS and NNOS pathways, it does not just accelerate local tissue healing. It creates a systemic environment that is more favorable to mitochondrial recovery and biogenesis throughout the body. Everything we have covered, from the inflammaging loop to the HPA axis blunting to the melatonin decline to the mitochondrial fragmentation, is not a collection of separate problems. It is a single integrated system that has lost coherence. Each layer amplifies every other layer. Each mechanism of failure becomes a trigger for the next. And everything we have discussed up to this point has been pointing toward one conclusion, which is not simply that the problem is complex. The conclusion is that because the problem is integrated, the solution must also be integrated. A protocol built around a single target will fail. A protocol that addresses the cascade, in the correct sequence, at the correct depths, can restore the coherence this system has lost. That is exactly what segment 3 of this video will present. And the sequence matters more than any individual element within it. Let's bring everything together, because until you see these 3 layers as a single picture, the protocol will look like a list of interventions. Once you see them as a system, the protocol becomes obvious. Begin at the surface. You have a chronic, low-grade inflammatory state, inflammaging, that never fully resolves between recovery cycles. This is not disease. This is the predictable biological consequence of decades of accumulated cellular damage, processed dietary inputs, suboptimal sleep architecture, psychological stress load, and the gradual depletion of the enzymatic machinery responsible for producing SPMs, the specialized pro-resolving mediators that instruct inflammation to complete and disengage. The result is an immune system that is chronically slightly activated, producing cytokine signals at levels too low to make you sick, but high enough to impair everything downstream. Those cytokines, specifically IL-6, IL-1 beta, and TNF-alpha, move upward into the neuroendocrine system and make direct contact with the hypothalamus. They blunt the HPA axis. They flatten the cortisol awakening response. They disrupt the precise hormonal choreography that coordinates morning tissue priming, daytime metabolic flexibility, and the crucial handoff that happens in the evening, when the body needs to shift from cortisol dominance to melatonin dominance, from catabolic to anabolic, from action to repair. That handoff, the cortisol-melatonin transition, is one of the most biologically important events of every 24-hour cycle. And in most people over 40, it is partially broken. Cortisol lingers longer than it should. Melatonin rises later and lower than it should. The window of deep anabolic repair, the 2 to 3 hours of slow-wave sleep where growth hormone pulses, protein synthesis peaks, mitochondrial maintenance occurs, and neurological consolidation takes place, is compressed, fragmented, or physiologically impoverished even when the total sleep duration appears adequate. And at the base of this entire cascade, invisible to casual observation, the mitochondrial network has begun to fragment. The fusion-fission balance has shifted. DRP1 is chronically activated by the same TNF-alpha that started this whole sequence. The electron transport chain is developing leak. ATP production per unit of fuel is declining. Oxidative burden is accumulating. And the cellular repair processes that depend on adequate ATP, satellite cell proliferation, protein ubiquitination, cellular autophagy, DNA repair, are all running on reduced capacity. Now let me give you a metaphor that I think will make this picture permanent. Think of your recovery system as an orchestra. Inflammation is the percussion section. It sets the rhythm for the repair process. The HPA axis and melatonin pathway are the conductor and the tempo. They coordinate when each section plays, how loud, for how long, and when to resolve into silence. The mitochondria are the power supply to the entire concert hall, the electricity without which no instrument can produce sound at the volume required. When everything works, the orchestra performs. The piece has structure, intensity, resolution, and silence in the right places. The silence is as important as the sound, because recovery, like music, requires both the activation and the resolution. Now imagine that the percussion section has been playing at low volume continuously, never stopping, never fully resting between performances. The conductor's timing has been slipping. Tempo slightly off, some entries late, some resolutions never arriving. And the power supply has been running at 80%, causing subtle distortions in every instrument that no one has yet traced back to the voltage problem. That is what recovery looks like after 40 when these three systems have lost coherence. It's not silence, it's not noise. It's distorted music, functional enough to pass as performance, structurally incapable of producing the full composition. The protocol we are about to present in the next blocks is not a list of supplements. It is a tuning strategy for an orchestra that has lost its coherence. The sequence in which you address each layer matters enormously, because addressing the mitochondria before you address the inflammatory signal is like replacing the power supply without fixing the conductor. The music might get louder, but it won't get better. What follows in the next blocks is the exact sequence: what to do, when, in what order, and why the order is not negotiable. Pay attention to the structure of this protocol. It is where everything we have built over the last 30 minutes finally becomes actionable. The protocol begins before the supplement. It begins before the peptide. It begins with a decision that most people in this conversation will find counterintuitive. And that decision is to stop trying to optimize individual variables in isolation and start treating your recovery system as what it actually is: an integrated biological cascade that must be addressed in sequence, from the bottom of the hierarchy upward. That hierarchy is specific. Layer 1 is the inflammatory environment. You cannot meaningfully improve HPA axis function in an environment of chronic systemic inflammation. The cytokines will continue to blunt the axis regardless of what you do at the hormonal level. Layer 2 is the circadian neuroendocrine coordination, the cortisol-melatonin transition, the nocturnal anabolic window, the sleep architecture that determines whether the repair machinery actually activates. Layer 3 is the mitochondrial substrate, the raw cellular capacity to execute repair once all the upstream signals have been correctly issued. Address Layer 1 first, then Layer 2, then Layer 3. Deviating from this sequence produces partial results that plateau quickly. Here's what addressing Layer 1 actually looks like, mechanistically speaking. The primary driver of unresolved inflammation is not the inflammatory signal itself. As we discussed, that signal serves a necessary purpose. The driver of unresolved inflammation is the deficit in specialized pro-resolving mediators. SPMs, resolvins, protectins, maricins are the active off switches. They are synthesized from EPA and DHA, the omega-3 fatty acids, through enzymatic pathways involving 15-lipoxygenase, 12-lipoxygenase, and cytochrome P450 enzymes. These pathways are themselves subject to age-related decline and are particularly impaired by chronic oxidative stress. This means that simply consuming omega-3 fatty acids is not sufficient if the enzymatic machinery to convert them to SPMs is compromised. You need both the substrate and the enzymatic support. At the same time, addressing the NF-κB pathway directly, through compounds that have documented inhibitory activity at this transcription factor, reduces the chronic cytokine signal that is upstream of everything else in this cascade. This is a fundamentally different approach from general anti-inflammatory strategies. You are not suppressing inflammation broadly. You are targeting the specific transcription factor that is maintaining the chronic, unresolved inflammatory state. And before I continue, I want to say something directly and honestly. This channel is approaching a significant milestone, and if what we've covered in this video, and in the 3 parts before it, has changed how you understand your own body, I'd ask you to consider subscribing if you haven't already. Not because the numbers matter for their own sake, but because this work continues. Every protocol on this channel is reviewed by a clinical pharmacist and researcher. That process takes time, rigor, and resources. And the community that grows here is the reason it stays credible. So if this is landing, subscribe. It takes 3 seconds and it means this continues. Now, back to the protocol. Because what I've just described at the inflammatory layer is only the foundation. Layer 2, the circadian neuroendocrine coordination, requires a set of interventions that are more nuanced, more time-sensitive, and in many ways more impactful on the lived experience of recovery than anything at the inflammatory level. The reason Layer 2 is so immediately felt is simple. The cortisol-melatonin transition governs the quality of every single night's recovery. And for most people over 40, this transition is being disrupted by 3 concurrent and compounding factors that are almost never addressed simultaneously. Chronic light exposure in the blue spectrum range during evening hours, which delays melatonin synthesis by suppressing pineal activity through the retinohypothalamic tract. Elevated evening cortisol that persists past 9 p.m. due to HPA axis dysregulation and insufficient parasympathetic activation during the late afternoon. And insufficient dietary serotonin precursor availability, specifically L-tryptophan and 5-HTP, that limits the substrate available for melatonin synthesis, even when the pineal gland's enzymatic capacity is adequate. Each of these factors interacts with the others. Blue light suppresses melatonin, while simultaneously maintaining HPA activation. Elevated evening cortisol directly inhibits the enzyme HIOMT, which is the final enzymatic step in melatonin synthesis. Low serotonin precursor availability limits production regardless of pineal enzyme activity. These are not 3 separate problems. They are one problem with 3 contributing drivers. And the protocol must address all 3, in a specific sequence, to restore the cortisol-melatonin transition that makes deep anabolic sleep biologically possible. Block 8 will now lay out the complete specific protocol, dosage ranges, timing windows, sequencing logic, and the peptide interventions that support the mitochondrial layer once the upstream conditions have been correctly established. The complete protocol is organized into 3 phases, corresponding to the 3 layers of the cascade, and a daily timing structure that synchronizes the interventions with your circadian biology. Timing is not optional. The same intervention taken at the wrong time of day can be neutral or counterproductive. Phase 1. Restoring inflammatory resolution. Morning, with the first meal. Omega-3 fatty acids, specifically a high EPA formulation. The research supporting SPM production points to EPA as the primary precursor for resolvin E-series compounds. The dose that has demonstrated meaningful effects on SPM production in clinical studies ranges from 2 E1 to 4 milliliters of EPA per day. This is substantially higher than standard supplement doses. Use a triglyceride form omega-3 for superior bioavailability. If using an ethyl ester form, consume it with the highest fat meal of the day. To support the enzymatic conversion of EPA and DHA into SPMs, include magnesium glycinate, which is a cofactor for the 15-LOX enzyme pathway, and vitamin D3, which has documented regulatory effects on NF-κB activity and has been shown to upregulate SPM-related enzymatic pathways in several clinical trials. For direct NF-κB pathway modulation, bioavailable curcumin, specifically a phospholipid complexed form at 500 to 1, 00 mg, or a BCM95 standardized extract. This is not generic turmeric. The bioavailability differential between standard curcumin and phospholipid complexed curcumin is substantial, approximately 29-fold in direct pharmacokinetic comparison. Combined with boswellic acids, which independently modulate leukotriene synthesis and NF-κB, this creates a documented and mechanistically coherent anti-inflammaging foundation. Phase 2. Restoring the circadian neuroendocrine transition. This phase is structured around the evening window, beginning 4 to 5 hours before your target sleep time. At 4 hours before sleep, complete all vigorous physical activity, if any is planned. Cortisol generated by exercise takes approximately 90 minutes to return toward baseline, but the sympathetic nervous system activation, associated with high-intensity training, can persist for 2 to 3 hours and significantly impairs the parasympathetic shift required for the cortisol-melatonin transition. At 3 hours before sleep, eliminate all blue-spectrum light exposure. This is non-negotiable for this protocol. Use blue light blocking glasses rated above 98% blue light filtration, or switch to warm amber lighting. The research from Harvard's Circadian Biology Lab demonstrates that even low-intensity blue light exposure in the 2 hours before sleep can delay melatonin onset by 90 to 150 minutes and reduce total melatonin peak by 20 to 30%. No supplement compensates for this input. At 2 hours before sleep, L-tryptophan at 1, 00 to 2, 00 on an empty stomach or with a very small carbohydrate serving. The carbohydrate facilitates tryptophan crossing the blood-brain barrier by competing insulin-mediated amino acid transport. Alternatively, 5-HTP at 100 to 200 mg achieves the same outcome with a shorter pathway to serotonin synthesis. This provides the substrate for melatonin production in the following enzymatic steps. Magnesium glycinate taken at this time shifts its role from cofactor to nervous system modulator, specifically through its antagonism of NMDA receptors, which reduces neurological activation in the evening and supports parasympathetic dominance. At 30 to 60 minutes before sleep, melatonin. And the dosing here is specific. The research does not support the doses most people use. Multiple studies, including a landmark paper from the MIT group, have shown that physiological melatonin replacement in individuals over 40 requires doses in the range of 0.3 to 1 mg, not 5 to 10 mg. Higher doses produce a pharmacological rather than physiological response, oversaturate MT1 and MT2 receptors, and paradoxically shorten sleep duration by disrupting the normal decline phase of melatonin in the early morning hours. For individuals who require higher doses initially due to significant melatonin depletion, titrating up to 2 to 3 mg is acceptable. with the goal of reducing toward physiological range over 8 to 12 weeks as endogenous production is restored. All protocols on this channel are reviewed by a clinical and research pharmacist, because accuracy here is not optional. Phase 3. Mitochondrial recovery support. This phase operates on a longer timeline. Mitochondrial biogenesis and network restoration are processes that require weeks to months, not days. But the daily inputs are specific. First, CoQ10 as ubiquinol, the active reduced form, at 200 to 400 mg daily with the highest fat meal. Ubiquinol is a direct electron carrier in the mitochondrial electron transport chain. Its decline after 40 is documented and significant. Supplementation at these doses has been shown in multiple clinical trials to improve ETC efficiency, reduce markers of mitochondrial oxidative damage, and support ATP production in aging cardiac and skeletal muscle tissue. Second, PQQ, pyrroloquinoline at 20 mg daily. PQQ is one of the few compounds with documented evidence of directly stimulating mitochondrial biogenesis in humans, specifically through upregulation of PGC1-alpha, the same transcription factor connected to BPC-157's nitric oxide pathway discussed in Block 5. It also functions as a mitochondria-targeted antioxidant and has been shown to improve measures of cognitive energy and physical recovery in aging populations. Third, for those working with a medical provider and seeking the most advanced tier of this protocol, BPC-157 at 250 to 500 mcg daily, administered subcutaneously or orally depending on the primary indication. Its role in this protocol is specifically its documented upregulation of the nitric oxide pathway through eNOS and NNOs, its modulation of VEGF and angiogenesis for tissue recovery, and its systemically stabilizing effect on the gut-brain axis, which is not incidental, because intestinal barrier integrity is itself a major driver of the systemic inflammatory load underlying inflammaging. There is a reason gut health appears in almost every longevity discussion. The gut is one of the primary sources of the LPS, lipopolysaccharide, that chronically activates TLR4 receptors and sustains the NF-κB signaling we have been working to resolve throughout this protocol. This is not a protocol of maximalism. It is a protocol of sequence, specificity, and mechanistic coherence. Each element addresses a defined target. The order matters, the timing matters, and the foundation, sleep architecture and inflammatory resolution, must be established before the mitochondrial interventions can deliver their full effect. You started this video asking a question you couldn't quite articulate. You had the feeling, but not the language. You knew something was different, not broken, not sick, just different in a way that no one had fully explained. And somewhere in the background of that feeling was a fear that maybe this was just what this age felt like. Maybe this was the ceiling. What you now understand is that the ceiling was never biological. It was architectural. The ceiling was a system that had lost its coherence, not its capacity. The inflammatory signal that never fully resolved. The hypothalamic rhythm that shifted just enough to compress the anabolic window your body counts on every night. The mitochondrial network that fragmented gradually, invisibly, in increments no blood test was tracking. None of these are aging. All of them are mechanisms. Mechanisms have levers. And here is what I want you to sit with. Because this is the reflection that I think matters most, not as a conclusion, but as a beginning. The moment you woke up this morning and put your feet on the floor. That moment existed in biology before it existed in your awareness. Your cortisol awakening response had already attempted to prepare you. Your melatonin had already tried to extend its nocturnal window as far as your pineal function allowed. Your mitochondria had already worked through the night on the repair queue that the day left behind, doing what they could with the resources and the signaling they had available. Your body was not passive while you slept. It was working. It has always been working. The question was never whether your body wants to recover. It does. Every molecule in you is oriented toward repair, toward adaptation, toward coherence. The question is whether the environment you are creating, internally and externally, is one that allows that work to complete. Or whether you are handing your body a repair job every day and then removing half the tools before it finishes. You now understand the tools. You understand the sequence. You understand why sleep alone is not recovery, and why inflammation is not the enemy, but its chronicity is. You understand why the cortisol-melatonin transition is the hinge point that most health strategies miss entirely. You understand why the mitochondria are not a biohacking trend, but the literal site where recovery either happens or doesn't. And this is the part I want to leave you with, not as motivation, because this doesn't require motivation, but as orientation. The difference between recovering and surviving is not a matter of effort. It is a matter of coherence. Your body knows how to recover. It learned that process over millions of years of evolution. What changes after 40 is not the knowledge. It's the environment. The signaling becomes noisier. The resolution machinery slows. The circadian rhythm loses some of its amplitude. The mitochondria lose some of their network integrity. What you are doing with this protocol is not hacking your biology. You are reducing the noise so your biology can do what it already knows how to do. You are giving the orchestra the conductor back. You are restoring the power supply to the level that allows the instruments to play at full volume. Recovery is not something you add to your life. It is something you stop interfering with. That is the distinction. And once you feel the difference, not just read it, but feel it, in your body, in your mornings, in the quality of energy that comes from genuinely repaired tissue rather than compensated deficit, you will not go back. Now. Before you go. If this video reached something in you, if it gave you a framework you've been missing, or named something you've been feeling for years without the language for it, I want to ask you to do something specific, not for the channel, for someone you know. Share this video with 5 people over 40 who are living in the gap between what they know they should feel and what they actually feel every morning. Not the people who will dismiss it, the ones who are quietly ready for it. You know who they are. Send it without explanation. Let the content speak. And if you have a question or a topic you want us to go deeper on, leave it in the comments. Not a generic comment. A real one. Tell me what hit you. Tell me what you want to understand next. This channel is built from those conversations. We are here every week, reviewing every protocol, checking every reference. Staying rigorous so you don't have to take the information on faith, you can take it on evidence. The next video in this series is already in development, and what it covers will make everything in this video make even more sense in retrospect. You'll want to be here for it. See you there. All scientific references cited in this video are listed in the description below. Timestamps for each block are also provided. This content is for educational purposes only and does not constitute medical advice. Consult a qualified healthcare provider before initiating any protocol.